Well, thanks very much for the kind invitation and introduction. And I think I saw some Hare Krishnas down the road on their way here. And I want to thank Mrs. Smart, with whom I had the pleasure of having lunch, for the family's enabling this lectureship. So, the human bacterial arms race for iron. I give a fair number of lectures, pretty rarely to an audience like this. So I have tried to, which ranges, of course, from experts in chemistry, I think I can deal with that, to experts in medicine, I might have more trouble with that. So I have tried to arrange a quite general talk. And for the chemists, I hope you'll bear with me if I have some things that are a little simple. For the others who may find some of the chemistry a little not so simple, I've tried to keep the jargon down and maybe have a few extra slides to explain things. So, iron. Rudyard Kipling. Cold iron. Gold is for the mistress, silver for the maid, copper for the craftsman cunning at his trade. Good, said the baron sitting in his hall, but iron, cold iron, is master of them all. Right? I think he had something other and different in mind than what I'll be telling you about tonight. So iron is essential. You can't live without it, and with too much, you can't live. Essential functions of iron, oxygen transport, electron transport in cytochromes, oxygenases and other enzymes. How much iron is in us? Well, I'll show you it's about 4 to 5 grams of iron. And so whether or not one is against the death penalty, I'll put aside any ethical questions or political questions there. Why is cyanide a lethal poison? A lot of people will say, and I've read it in my local newspaper, that it's because people asphyxiate from iron. In other words, from cyanide. In other words, the cyanide is blocking the site on iron that would normally carry oxygen. Were that the case, 5 grams of iron is about 1/10 of a mole, if you remember your high school chemistry, right? So that would mean that you need a lot of cyanide to kill you. And in fact, it takes parts per billion. That has nothing to do with oxygen transport. It interrupts electron transport in cytochromes. You've got a lot fewer of those molecules around, and they're essentially the energy transport system for human metabolism or other mammalian metabolism. So, toxic effects of excess iron. I'll give you a few examples of that in this introductory part of the talk. Free iron gives rise to free radical or Fenton chemistry. And so this is associated, for example, with inflammation. I think it's becoming increasingly clear that a lot of inflammatory diseases have a key role of iron regulation in the inflammation that is part of the disease process. Iron is required for infections by pathogens. If you have a bacterial infection, for one bacterium to turn into 2, it needs twice as much iron. Guess where it gets that iron from? You as the host. Okay, so that's much of what the central thesis of this talk is about. But first, a little general background about iron distribution. You only need about 1 to 2 milligrams per day if you're an adult male. So you have 5 grams of iron in you. That means that every iron atom in your body has an average lifetime of thousands of days, right? So we're extremely efficient at recycling iron. We don't regulate getting rid of excess iron. We have no mechanism for that except blood loss. The only way we regulate the amount of iron in us is by uptake. The 4 to 5 grams of iron excuse me, in the body About 70%, I mean the 68% is a lot more precise than the actual number. The ranges here are fairly large. 70% circulates as hemoglobin in blood cells, about 25% is stored in the liver, about 7.5% is stored as myoglobin. So myoglobin is stored over here in muscles. Again, one of my favorite questions to freshman chemistry students, and I'll use you folks as a foil for this, is whales. Whales can dive down to 3,000 feet, and that's 100 atmospheres, right? And at 100 atmospheres, their lung volume is 1/100th of what it was at the surface. So, and they can stay down for certainly 10 to 30 minutes, depending on the species. So that means they need a lot of oxygen. If that oxygen is in their lungs, they're floating like a cork on the surface and sinking like a stone at 3,000 feet. How do the whales do it? Well, the oxygen, they don't store the oxygen in the gaseous form. They store it as myoglobin in muscles. And so it's now liquid, it's incompressible. And so their buoyancy almost changes not at all. They exhale completely before they do deep dives. Okay? So iron recycling and excretion. And again, so dietary iron comes in. It's absorbed in the duodenum. Iron storage is primarily in ferritin, ultimately inside cells. Ferritin I've described as rust with chicken fat around it. It's a little particle of about 4,500 iron atoms. Okay, and it really is just ferric oxide hydroxide, so it's rust. And the growth of the particle is arrested by coating it with the protein. And so you can have up to 10th molar iron in ferritin, high concentrations. Whereas if you have just free ferric iron, you put it in buffered solution, it precipitates out as rust. Okay? Ferritin is a very old protein. It's found in plants, animals, bacteria, even archaea bacteria. So the solution of that iron storage was solved at a very early stage of evolution. Okay, 1 to 2 milligram per day is excreted by various means. Most of that is bleeding. So menstruating women or pregnant women need a lot more iron. Some of it is hair loss. For some of us, that's more of a problem than others. But again, these are small numbers, right? You have a lot of iron, and you don't need to absorb much of it. Transferrin is the way we move iron around. It's an amusing name, right? When they assigned it. So it's in our serum, and it shuttles iron to and from iron storage sites. Okay. It's a molecular weight 80,000 protein, and it has 2 iron binding sites. So this is like designing a huge freight train to carry 2 passengers. Okay. Works for us because we recirculate this over and over and over again. One of the things I want to show you is that for bacteria, they cannot afford to make a huge molecule like this because most of the time they will use that molecule once and it's gone. So they have to make small molecules to transport and uptake iron. So the transferrin cycle, it's a protein that binds these 2 iron sites. When both sites are bound, it binds to a receptor. Binding to that receptor triggers an invagination of the cell membrane. And then inside the cell, the pH is dropped. It becomes more acidic. The iron is released. The released iron is eventually processed, and the protein is recycled into the serum again. Okay, so that's the process for uptake and transport of iron as mediated by transferrin. Iron uptake is mediated across the intestinal epithelium. So the epithelium is diagrammed here. So this is the way we absorb the nutrients in our food. the iron is reduced from iron 3, which it is, anything you eat is going to have iron in the higher oxidation state of ferric iron because of oxygen being around us. But it's reduced to ferrous ion, which is a lot more soluble, and then there's a transporter that transports specifically ferrous ion across the apical membrane, and then iron is stored, as I told you earlier, as ferritin after being reoxidized from ferrous ion to ferric. Okay? So there's a redox cycle that's part of the transport process, and nature, of course, is as good an engineer as she is a chemist, so dividing up all of these compartments of the cell is an important part of the transport and storage. Beta thalassemia. This is a terrible disease. It's a genetic disorder in hemoglobin synthesis. So you have 4 proteins associated with hemoglobin, 4 protein coils. And an error in the synthesis of one of those is associated with this disease. Now you might ask, why would a disease, an hereditary disease, live in a population over very long periods of time? You would expect, if it's lethal when you get 2 copies of the gene, you would expect that over time that the survival would mean that you'd mutate away from having that gene survive in the general population. But in this case, there's a selective advantage. If you have 1 copy of the gene, it gives you partial protection from malaria. Okay? And so in parts of the world where malaria is a terrible killer, over this long period of time, human populations in those regions have evolved to this point where there's high populations of this genetic error present. So in some regions, thalassemias can reach genetic frequencies of 70% or more. Okay? And of course today, people from Southeast Asia or from the Eastern Mediterranean, they come to the United States, their children, their grandchildren, these become serious diseases for not just places like Greece, but for the United States. How do you treat the disease? Well, you get bone marrow expansion in a beta thalassemia patient. This is to show you that the effects of this are quite terrible. The treatment of the disease is whole blood transfusion. So you have a patient whose blood is not working. What do? Well, you give them a transfusion of somebody's blood that does work. Well, that's terrific, but what have I told you about how much iron you need per day? Every unit of blood is about a year's worth of iron. And so these patients develop a terrible iron overload. And that's what's lethal about the disease. So supportive treatments, blood transfusions. There is one drug that is approved. There's a second one on the way that's an iron chelator that can be used. This is terrible treatment. It's young people, typically young adolescents, who typically show the first onset of this disease. They have to walk around with portable peristaltic pumps, shoving this stuff into their arm 24 hours a day because the clearance time of this compound is very short. So hemochromatosis, totally different disease, but also related to iron. Hemochromatosis is a mutation of one particular gene. It's the most common hereditary disease in Northern European descent. 1 in 8 Americans are carriers. 1 in 200 have the double mutation. 1 in 4 Irish are carriers. 1 in 16 have the double mutation. Disease characteristics, typically the onset here, instead of being as children or as adolescents, it's later in age. It's 40 to 60. The accumulated iron produces tissue damage. Target organs are the liver, heart, pancreas. Symptoms, what's the treatment? Phlebotomy, a fancy name for bleeding. Okay? And so people who have this disease will go in regularly, essentially giving blood, right? And so that clears the blood away from enough blood away from their system so that they don't have iron overload. Okay, so now we come to the human bacterial arms race for iron. From Shakespeare's King Lear to treat the Earl of Gloucester's eyes, he's just had his eyes gouged out, poor man. His aide says, "I'll fetch some flax and whites of eggs to apply to his bleeding face." Of course, this was long before anyone knew anything about bacteria or bacterial infections. But it had been observed that egg white retarded infection. We know today that's because egg white has a large amount of a protein called ovotransferrin. If you think about it, an egg is laid in a pretty non-sterile environment, right? And yet, a bacterial infection of the egg takes a long time to develop. And the reason is that the white of the egg has this protein that keeps the iron concentrations down so low that bacteria can't grow. So, the second example, in the 1850s, Dr. Trousseau, he was a professor of clinical medicine who was in charge of a tuberculosis clinic. And of course, in those days, if you were sick, you needed dietary iron supplement. You had weak blood, right? And so he was a careful observer, good scientist. He noted that his patients who got dietary iron supplements became ill faster with the tuberculosis than those who didn't. And we know today that's because the organism that causes the tuberculosis needs iron to grow. And so you're feeding the organism with the dietary iron. So, how much iron do the bacteria need? Roughly, on a per weight basis, they need about the same amount of iron as we do. Okay? It's an essential nutrient for virtually all living organisms, certainly for all human pathogens. It's involved, again, in oxygen metabolism, electron transport, a number of processes that are essential for life. Now, in the Earth's crust, it's the fourth most abundant element, but it's very limited bioavailability. So this little equation I'm going to have to translate for you a bit, but that at neutral pH, the equilibrium concentration of ferric ion is 10 to the minus 18 molar. That is a very small number, okay? So iron is profoundly insoluble because rust is so stable, okay? So all bacteria produce and secrete siderophores, sideros, the Greek word for iron, phoros for carrier, and now we come to my 5-minute summary of the evolution of the planet. So the earliest record of life on Earth is, depending on who you talk to, is about 3.5 to 4 billion years ago, okay? And so virtually all life for the first billion years was anaerobic. The atmosphere was a reducing atmosphere there, and so iron already had been incorporated is in all kinds of important biological processes, and it was widely available. You could have up to a 10th molar, high solubilities of iron in the oceans as iron 2, as a ferrous ion. Okay? So then the cyanobacteria, about 3 billion years ago, started photosynthesis, and as a byproduct, they made this noxious stuff called oxygen, right? And so what you see is over a period of about 300 million years, the oxygen levels go from essentially 0 to modern levels, okay? And associated with that, the reason it took a long time for that process to happen was that all of the methane in the atmosphere was being oxidized, and all of the ferrous ion in the oceans and other oxidizable species were being oxidized. And you can see this in the geologic record. You see layers of precipitated ferric hydroxide, okay, that correspond to this geological time. So now at this point in time, of course, only microorganisms exist, right, single-cell organisms. So you're hooked on this stuff called iron, and suddenly it's not available to you. And by the way, of course, this period of time probably corresponded to the greatest mass extinction in the Earth's history because almost all of the anaerobes died at that point. So the ones that didn't had to arrange to take up, store, transport iron in this new oxidation state, +3. And so this is the only slide about coordination chemistry explicitly, okay? So I've got a metal ion, in particular iron 3 is never naked. It's always surrounded by some atoms that are directly bonded to it. In general, for all of the siderophores, all of the molecules I will talk about today, there are 6 atoms that coordinate in a more or less octahedral arrangement around the metal center, okay? And what I want to show you is that if you connect these atoms pairwise, you make something that's like a little blade on a 3-bladed propeller. And I'll show you later, that's chiral. That's got a chirality associated, left-handed or right-handed, okay? So that's the reason why propellers, when you turn them one way, push the boat forward, and you turn it the other way and they go backwards, right? So there's a twist angle. This is looking down the threefold axis that has trigonal symmetry of that tris bidentate complex, okay? So how important is iron to bacterial growth? Yersinia pestis, which caused the Great Plague, the bubonic plague, Yersinia pestis, produces Yersinia-bactin. This is a molecule that has a number of coordination sites. Now I showed you that the iron has to have 6 coordination sites coordinated. Now it doesn't have to have all of those coordinated by the same molecule, but the most stable complexes will be one in which the molecule satisfies all 6 of those coordination sites. Otherwise there may be water there or some other atom or molecule coordinating to the iron. So the bacteriostatic effect of serum is blocked by addition of an iron complex. If you add a chelator, ferrioxamine, which I'll show you later, the LD50 is the lethal dose for 50% of the population in mice, okay? For Yersinia pestis, the LD50 in mice is normally 10 to the 8. That is, it takes 10 to the 8 cells of the bacterium before half the mice are killed. And the reason is their immune system is protecting them from that bacterial infection. If you now add this complexed iron, that number drops from 10 to the 8, 100 million, to 10. There's a 10 million-fold increase in the pathogenicity of the organism because of the presence of the chelated iron. I think that's an impressive number, right? So, bacteria and their siderophores. These are just some examples, and there's not going to be a quiz about the names. Mycobacteria tuberculosis, which causes tuberculosis, produces these 2 siderophores. So these siderophores are low molecular weight iron chelators, like the Yersinia-bactin I showed you, that the bacteria make specifically to complex iron. And they're highly selective. They complex iron, ferric iron almost exclusively and very tightly, okay? Pretty good trick for a little molecule, right? That takes us a big protein to do. Salmonella produces, the organism that causes salmonella produces enterobactin and salmonellin. E. coli, which we need to digest our food, but if you get the wrong variant in your hamburger, you're in trouble, right? Bacillus anthracis, you really don't want this, causes anthrax, and it produces 2 siderophores that we characterized a few years ago, and I'll talk a little bit about later. Pseudomonas aeruginosa, it doesn't cause cystic fibrosis, but it's an opportunistic infectious agent that is typically what causes the biggest problem for patients with this. I told you about Yersinia pestis, which causes the plague and uses Yersinia-bactin, and Vibrio cholera, which causes cholera, produces a siderophore called Vibriobactin. So I've been doing this chemistry for some time. My late colleague, Joe Nealens, was in the Berkeley Biochemistry Department. In my view, he's one of the two starting heroes in microbiology and biochemistry to really characterize the iron transport processes. So this all started in 1952. These molecules had been isolated. There was no understanding that they were involved in iron transport. But starting in the late 50s, early 60s, it was discovered that they were involved in iron transport. Excuse me. And the molecule at the lower left is enterobactin. And in each case, the atoms that coordinate to the, well, I guess I didn't highlight this one. And the atoms that coordinate to the iron are highlighted in red here. So Neilands had first characterized ferric ferrochrome, and the crystal structure was done in the late 60s. I had just arrived as a brand new assistant professor at Berkeley in inorganic chemistry, right? And I did something that at the time was highly unusual. I invited a biochemist to give an inorganic chemistry seminar, okay? That wouldn't sound so strange today, but believe me, it was out there then. So this is the structure. This is a ring, a cyclic ring. It's got 6 amino acid groups strung together. 3 of them are glycine, and then there are 3 ornithines that come up. And they terminate in hydroxy groups. This is an amino hydroxy group that's bonded to an acetyl fragment with other spinach on it. The point is, this is a hexadentate chelator. The structure is a little bit like a 3-legged piano stool. So you've got the chair, which is up, and it's upside down. So you've got the chair and then 3 arms that come up. And those 3 arms have 2 iron binding sites each, and so it wraps around the metal. And what I've shown over here is the view down that, remember that 3-bladed propeller that I was talking about? This is the view down what would be that 3-bladed propeller. And it's left-handed, right? And so at the end of Neilands' talk, I said, does it make any difference to the organism whether it's left-handed or right-handed? And he said, I don't know. I don't think anybody ever thought about that. And I was off and running, okay? So it's a very dangerous thing for an assistant professor at a place like Berkeley. I took about 6 months off and did nothing but reading because I knew no biology at the time. And then for about 10 years, I was the only chemist working in this area, which is even more worrisome because there are at least 2 possible reasons why you're the only one doing something. But it worked out, right? So another person I want to show you is Roe Byers at the University of Mississippi Medical School. He's a real card-carrying microbiologist, and he's worked on a number of important systems. In particular, he has a laboratory where he can work with and isolate Bacillus anthracis. He sent us sterile samples of the metabolites from this, and from those sterile samples, this was when I first talked about importing products of Bacillus anthracis, my NIH, well, the administrators on the Berkeley campus got very excited, but we convinced them that these were sterile samples. And so we could purify and characterize the siderophores. So the siderophore structures I'm going to talk about today are shown here. Again, there's not going to be a quiz. Enterobactin, which is kind of the Arnold Schwarzenegger of the siderophore field. And the reason that's important, and one of the early things that my research group got involved with, is that one of the ways bacteria compete with one another is my siderophore is bigger and badder than your siderophore, okay? So if you can bind iron much more tightly than another siderophore, you can outcompete the microbe that is producing that siderophore. Now that's a little simplistic, but by and large that's the case. So this has a huge stability. Samachelin has, it has 2 glucose groups on the enterobactin. Why put spinach on Arnold Schwarzenegger, right? Why add this additional complication to a structure that is already so enormously stable. Bacillobactin is made by Bacillus anthracis. It looks a lot like Enterobactin. It's got the same ring. It's got a glycine spacer in the middle. This is for the chemists. But the same catechol groups. There's three bidentate, as we say, that's two-toothed groups that bind to the metal. Aerobactin, this is a citrate derivative that's got two hydroxamate groups. So it coordinates to all of the 6 coordination sites of iron. And it's produced by E. coli, like Enterobactin. But this is a much weaker chelator. But what puzzled me for 20 years was the E. coli that you need in your gut makes this. The E. coli that you don't want in your hamburger makes this wimpy chelator. What's with that? Okay. So carboxymycobactin, this is made by, among other things, the organism that causes tuberculosis, and petrobactin, which is the alternative siderophore of Bacillus anthracis. So, put yourself in the position of the bacterium, watching all these molecules float by. How do you know which ones have iron in them? Okay. And the answer is, if it looks like this, there's iron in there. So this is a diagram of gram-negative bacteria. They have 2 membranes. So this is the outer membrane. Out here is solution. Here is the cytoplasm of the cell. And this area in between is called the periplasm. And resident on the outer membrane are all these receptors. There's a receptor FEP for ferric enterobactin. And it recognizes the catechol iron complex part of the molecule. You can chop away, and we did that by making similar molecules, you can cut away the bottom part of the molecule, as I'll call it, and it still gets recognized as long as you have that iron catechol part. There's a ferric citrate receptor. So just citric acid complexes iron, and there's a protein receptor that recognizes that ferric citrate, and on and on and on, okay? So the problem with that mechanism, of course, is that if you're going to utilize an iron siderophore or other complex, you have to specifically make a receptor for that. And the interesting thing is that for E. coli, for example, and most other organisms, it will have receptors for siderophores it never manufactures. So it coasts along on the hard work of other bacteria. Okay? So chirality and recognition. Remember that 3-bladed propeller I talked to you about, right? So this is left-handed. This is a left-handed screw. Okay? And here's rhodoterilic acid. The top and bottom of the molecule are here. So there's an iron there and an iron there. And I don't know if you can see this, but it's a right-handed screw at the metal center. Okay? And what I did not point out sufficiently is that ferrochrome is a left-handed screw. Remember that structure that Neilands had done? That has a chirality to it, a set chirality, and it's left-handed. And so the question that we addressed, and this is an old paper, but it's the nicest example still, the question we asked was, does the recognition of that iron ferrochrome depend on the chirality at the metal center? Does it depend on whether it's left-handed or right-handed? So we gave it rhodoterilic acid, which is the opposite chirality at the metal center than ferrochrome is. But again, I told you that it's only this part of these molecules that are recognized. The other stuff down here doesn't matter. And what you can see, so this is uptake of radioactively labeled iron as a function of complex concentration, and it's a terrible transporter of iron. Then we had some Japanese colleagues, organic chemists, who had made the mirror image molecule of this. Never before seen on the planet, right? It's an artificial compound. We made the iron complex of that, which now, if this is right-handed, it must be left-handed. Okay, you staying with me? Sorry, it's a little complicated, but okay, so it's left-handed, and it is very effective at delivering iron. And so this proved that the chirality at the metal center was crucial for the biological transport. Okay? So I think I will skip that one. This is, given the response to the last question. FEP-A is the protein receptor for ferric enterobactin. Here's the structure of the metal complex. So here's the iron down here at the business end of the molecule with its 3 catechol groups around it. You can see it's kind of a little octahedral coordination, right? There's 6 oxygen atoms around the iron. And this is the triserine ring up here in these carbonyl groups. And it's this part of the molecule that gets bound to the calyx of the protein. Now, most of this protein is just pipe because it's got to go all the way through the outer membrane. Okay, so it has to be long enough to go from outside the membrane, from the outside of the cell, to the inside of the cell. The action is up here where this molecule gets bound. So it requires recognition. It's an energy-driven process. And this is typical of all of these transport proteins. They are what are called beta barrels, but it's essentially nature's version of a pipe that spans the membrane. And the variation in this is the receptor group at the top that is tuned to recognize a specific iron transport molecule. Gram-positive bacteria. If they're gram-negative bacteria, it's logical that they're gram-positive bacteria, right? So the bacterial world is divided largely into these 2 families. Gram-positive bacteria have only this one thick membrane. The cell wall is greasy, waxy stuff, and then the membrane itself is inside that. Okay, much less is known about iron transport in gram-positive bacteria, both from what my group has done and from others. And yet, many of the most important human pathogens are in this class of organisms. Okay, so one of the things that is on our list of current goals is to try to understand this better. These are much harder to work with in general. So active transport at the cytoplasmic membrane is required, and as I said, less is studied. So Bacillobactin, gram-positive, and Enterobactin, gram-negative, are shown here. These look pretty similar, right? Even to a chemist, these look pretty similar. So the differences are highlighted in red. So you've got these catechol groups that bind the iron, and the difference is that Bacillobactin is not made from serine, it's made from threonine. That means it has an extra methyl group at these positions marked in red. Looks like a pretty small change. The professional organic chemist would say, you don't understand, that makes all the difference in the world. And they're right. So here's the difference in structures. Okay, here's ferric enterobactin. We've seen this already when I showed you the receptor protein slide. This whole ring structure has now been turned inside out in Bacillobactin. And the reason for the extra spacer now becomes clear, because you need that extra length to go down and be able to completely encapsulate the iron. And what's really weird is that this is primarily right-handed. It's a right-handed propeller, and this one's left-handed. And the protein receptors for these guys, in general, distinguish between that. Okay, so they're gram-positive receptors for this molecule that will not recognize this one. Okay? So, immune response. So here we have this challenge, and it's a serious challenge. Again, more numbers. This is like pH. But let me just summarize it by saying your transferrin keeps free ferric ion concentration down to a small number, 10 to the minus 23, roughly. Okay? Small number. But ferric enterobactin has a concentration of free iron of 10 to the minus 35. It's 12 orders of magnitude more stable. So if you've got a bacterial infection in serum, or it's exposed to serum, the transferrin has no chance. It's going to release its iron to the chelator. Okay? Not good for us. And yet, very often usually that doesn't happen, and the question is why. So now about 9 years ago, a little more, I got an email from a colleague at the Hutchinson Cancer Research Center in Seattle, Roland Strong. He's by training an endocrinologist, so he works on the immune system, but he had morphed into being a protein crystallographer, okay? And he had isolated a family of proteins that he thought perhaps were binding siderophores. And he calls me up and he says, do you have any ferric enterobactin? Yeah, maybe close to the world's supply. We can. So we started a collaboration, okay? And this was the first published result of that collaboration, the cover of Molecular Cell. Not bad. So what was exciting was that this was the first evidence that the human immune system is playing an active role in disrupting siderophore-mediated iron transport. This protein binds siderophores and their iron complexes. So it's a kind of vacuum cleaner to keep our system free of the siderophores. And it's not subtle. These complexes, so the siderophore iron complexes in general are bright red in color. And so all you have to do is look in the microscope at the crystals, and you can tell, has it picked up ferric siderophore or not? If it's colorless, it is not. If it's bright red, it has. Okay? So, the protein doesn't just recognize one or two siderophores, as I showed you the microbial iron receptor proteins do. It recognizes a whole range of different chelating agents, siderophores, for iron, okay? So it recognizes enterobactin from E. coli, bacillobactin from Bacillus anthracis, carboxymycobactin from the tuberculosis organism. So pretty exciting stuff, okay? Now, one of the things, if you don't remember anything else from today, the one thing I'm going to claim credit for in recent times is the word stealth, okay? So siderophore stealth. So I told you that there was this weird business where enterobactin, which is this Schwarzenegger-like complexer of iron, is made by the form of the microorganism that is our symbiote, okay? So we developed an assay for the binding of siderocalin, this protein, binding ferric siderophores. And so what you see is aerobactin, which is the product of that nasty form of E. coli, is not bound by siderocalin. It gets under the radar of our immune system. It's a stealth siderophore. And this is stuck in the literature. I've now got about 24 references on this, so it's going to be there. Bordetella pertussis makes alkalogen. We characterized this molecule some time ago and its iron bonding characteristics. This is the organism that causes whooping cough. Okay, and it gets the iron it needs to grow by making this. Okay, again, it's not bound, and so you have this pattern that the super chelators that are made are bound and inactivated by siderocalin, and so to get around this, the microorganisms, the pathogens, have evolved to make these weaker chelators. They're not nearly as strong, but they're not bound by siderocalin. So samochelins, I showed you this parent molecule. So this is enterobactin that has two glucose groups stuck on it. Remember the question was, why put spinach on Arnold, right? And the reason is that this is now big enough so that it doesn't fit into the protein calyx anymore. And so the bacteria that make this form of enterobactin are super pathogens. They can be in the serum and they still act to deliver iron. Okay? Bacillus anthracis. Okay, you really don't want this. It essentially has two parts of its life cycle. Right? It can live in the soil for years as spores, okay, even decades. And at low temperatures, it primarily makes the super iron complexing agents, the bacillobactin. When it gets into its unfortunate host, and the temperature goes up to 37 degrees centigrade, it starts to make another siderophore, the one I showed you earlier, petrobactin, which I'll diagram again. And that's a stealth siderophore. So here's bacillobactin. It's a 2,3 catechylate that is a substitution. Here is next to these two oxygens. This may not look like a big difference, but by moving these two oxygens around one carbon atom in this ring, you put the angle of attack of the iron off at an angle so that it no longer complexes, and it doesn't fit into the calyx. So this is a model of the computer-generated diagram of the crystal structure of siderocalin in its pocket. And it's a little complicated to see here, so it's summarized here. Here's the dotted line represents the lining of the protein cavity, and the 2,3 catechol amide, like enterobactin, fits within that. The 3,4 catechol amide does not fit within that. Okay? And this crystal is red, and this crystal is white. Okay? So how general is this whole thing? I think it's quite general, and time will tell in the next many years, but we see a quite general pattern, E. coli. So in each case, the siderophores that are in red are stealth siderophores. The black ones are bound by siderocalin. Okay? So I think this is going to be quite a general phenomenon for all of our bacterial pathogens. Okay, so siderocalin defense in chickens. Protein, these have not been called siderocalin. You know, when you name something, it can be controversial and everybody objects. And I wasn't involved in naming siderocalin directly. But there's a protein found in chickens that looks very much in function like siderocalin. Now it has some structural differences. And there's another family of proteins that are found in worms. And from my perspective, there's a whole range of siderocalins that are produced by different organisms, everything from worms to human beings. Okay? But there's a difference in the recognition, and I won't go into the difference into the details of this. But in each case, the protein that is produced by these different organisms has a slightly different recognition pattern for the siderophores they recognize. Now, if you're a chicken and you're exposed to a different group of pathogens, the most dangerous pathogens, you know, what do you want to interrupt? Not necessarily the siderophores that humans interrupt. And so I think this is an evolutionary response, and I think we're going to see in the future a whole range of siderocalin type proteins produced in different families of organisms, but it's quite wide. As I said, it's all the way from worms so far to human beings. Siderocalin-9 transport. So the most recent, and we're getting close to the end here, okay? So hang in there. So what's emerged in recent years is that siderocalin isn't just a vacuum cleaner for siderophores and iron transport. Transferrin knockout mice still undergo organogenesis. What's that mean? Transferrin is involved in iron transport, and yet mice that have the genetics removed for the making of that protein can still generate organs utilizing iron. Somehow they're getting iron transport even though the transferrin uptake process has been shut down. Siderocalin is expressed about 10 to 500 times more in serum and urine after damage stimuli. Okay? So there's an active response that somehow if tissues are damaged, siderocalin is produced. That doesn't make too much sense when you think about siderocalin only as a vacuum cleaner for the siderophores. So are there components in the serum or urine that are cofactors? And the answer to that is yes. So this is a paper from just last year. It's pretty exciting. Catechols are human siderophores. Simple catechols. You can measure concentrations of, and I think that's on the next slide. So siderocalin requires a siderophore cofactor to bind iron. What's that mean? Unlike transferrin, siderocalin does not directly bind iron. It binds the molecule that has complexed iron. right? So that's what I mean by cofactor. And even just, so here's enterobactin, this is another name for it. Here's a range of catechols, including the simple unsubstituted catechols. This iron mobilization in components of urine is shown here. So no, the iron by itself is not mobilized, okay? You have to have it complexed, and then siderocaline will bind it and mobilize it. Oops. So I wanted to say one more thing about this. So the iron the catechol concentrations in human urine are about 200 micromolar, okay? And most of that comes from metabolism, from breakdown of things like enterobactin in our digestive system. Okay, so to me, this is quite remarkable. I think it's, for me, very exciting. This whole story, which I've been now working on for 35 years, has been like a terrific mystery novel. Every time you think you've got the plot figured out, there's a new twist and you're off running in a new direction. So I've had NIH support for this for now 38 years. And you might say, gee, how could you be doing anything interesting that long? I have a slightly more favorable view that I'd like to suggest to you. With the NIH having a success rate of about 20% for renewal grants, this is kind of like going to Las Vegas and rolling the dice successfully more than a dozen times, okay? So with that, I will close. I hope I've shown you that the human bacterial iron race has been an evolutionary result, that our major iron is a crucial factor for virtually all bacterial infections, and that and defending against that iron uptake is a crucial part of our defense against the infection. And remember the word stealth. Stealth siderophores are a way that the bacteria use to get around our immune system. And I want to acknowledge Dr. Roland Strong, these colleagues at the University of Argonne who are involved in other protein structures, students in my group, Benjamin Allred, Alison Sia, and Dr. Tetsuya Fukushima, who's a microbiologist that's brave enough to hang out with the chemists. And I want to thank you very much for your attention. Yes. I think you're supposed to wait for the microphone. We have a protocol here.
Thank you for a very engaging lecture. Pensacola, some call we're a large, small town, but we still have 3 huge cancer treatment centers here. So cancer is a big deal in Pensacola. We know that cancer is anaerobic. It hates oxygen. But at the same time, we want to prevent cancer, maybe even treat it naturally. But at the same time, we don't want to have excess oxygen to promote what you're talking about here. So what do you recommend for those of us who are natural health enthusiasts and trying to stay cancer-free when it comes to oxygen or an iron?
Well, there are all kinds of studies that show that to the extent you can keep free radical generation down, so antioxidants are good for you. You know, eat your beets, eat ascorbic acid. I think one of the interesting things about cancer is that neoplastic cells, cancer cells, have about 1,000 times as many transferrin receptors on them as normal cells do. And so because these are wildly growing, they're taking up the nutrients that they need to grow. And I'm not the first to suggest this, but it provides you with interesting ways that you might specifically attack the cancer cell as opposed to normal cells.
Carl Boher, University of West Florida. Could you elaborate a little bit more on ways in which the stealth siderophores could be perhaps treated? You have an understanding now of how these sneaky bacteria work. But after 35 years, do you have any ideas as to how physicians could use this knowledge other than giving antibiotics?
Yeah. So there's been a lot of thought devoted to, can you make antibiotics that have a kind of Trojan horse, right? So you have the siderophore attached. And I know that, so there's a group at Notre Dame that's close to having something with a pharmaceutical company on this. Marvin Miller is the organic chemist. But it took him 20 years. And it's tough because one siderophore isn't used by just one microbe, and you want a targeted approach. You don't want to, kill everything in there. As a general answer, I would say the more you understand about it, the more options you have for developing new treatments. Is there a specific magic bullet for a specific bacterium as yet? No. I mean, I think, if you want to keep away from Bordetella pertussis infection, make sure you inoculate your children.
Thank you.
Patty Krakowski. I have a girlfriend who has restless leg syndrome, and her doctor has told her she needs to take iron supplements. And I don't know if you have any opinion on that or not.
I think her doctor probably knows best. I presume he has done an analysis of her iron. So it used to be that to do a liver biopsy or liver analysis of how much iron you have required you to go in and snip out a chunk of liver. These days you can walk into a magnetometer and they measure how much iron is there. So if she really is anemic, there should be a measure of that. And if she isn't certain about that, she should go for a second opinion.
Yeah, well, since the iron is so important in these diseases, does that suggest that these folks that are wearing magnets around are up to or have a good idea?
To get any effect, they would have to have a magnet around them that is about 1,000 times stronger than an MRI magnet. Okay, so it's the energy, and so it's may make them feel good, but there's no way that's going to do anything to the iron.
All right, let's thank our guest.